Good afternoon. Thank you for joining the second half of the Steenbock lecture that Professor Jeff Gordon will give today. So it's my distinct pleasure to introduce Jeff. Jeff is the Dr. Robert Glaser, Distinguished University Professor. He's a professor in the Department of Pathology and Immunology at Washington University. And he's also the director of the Center for Genome Sciences there. And yesterday, Snehal did a fantastic job introducing Jeff, introducing the accomplished scientist and professor, and sort of telling us, putting forward sort of like the all the awards that Jeff done and his contributions to the field. Today, I'd like to give a more personal perspective of my time, sort of working with Jeff. I had the fortune to spend 6.5 years, six wonderful years in Jeff's lab, first as a postdoc and then as an instructor. And I sort of would like to talk about Jeff, the mentor, because he's a phenomenal mentor, one that is supportive, generous, and patient with his trainees. And that he challenges his trainees with these big ideas. And you saw yesterday some of the ideas that his lab is, what sort of goals that his lab is going after. But as big as the ideas is the levels of support that he provides, the trust, the resources. So trainees really flourish in whatever project, whatever going to the moon project that sort of they are pursuing. And beyond sort of the time in his lab, he really sets up people for success, and really, and that is sort of documented with the many of us that are around the globe, sort of at universities with faculty jobs. He equips really his trainees with with the latest technology to address these challenges, questions that he's going after, and he really makes these bold investments that are needed to develop new methods, new technologies, new model systems. He talked about the germ-free pigs yesterday, to set up, sort of his trainees with in a path to success. I think he's always 5 to 10 years ahead of the curve, and or at least ahead of me. So, he also has this amazing ability to motivate trainees and transmit this infectious optimism that he has. And as much as I miss all the fancy toys that we had in the lab, I really miss interacting with Jeff because he's sort of a positive source of energy, sort of positive, emanates positive vibes when you talk to him. And then that sort of takes me to the last the other aspect that I feel it's hard to describe without getting too personal, but I think it has to do a lot sort of with his success, and it's how he interacts with people, in his lab, and the superb environment that he really creates in the lab. And so you see this picture here that has about 30 people, about a third of those people, about 10 or more, have been with Jeff for 15, 20, some of them 30 years. These are amazing people, really talented people. Talk about family, talk about sort of lab family, trust. People love really working in with him. And that is sort of really transmitted. I felt like that was really transmitted as me as a newcomer into the lab. You join this family, with this, family of cheerleaders that are working, there to support you and help you succeed. That was a really amazing experience. And this, sort of pursuing these challenging projects, but in this, supportive environment where people are really inspired to achieve. So thank you, Jeff, for the inspiration, and and for coming to Madison and giving us 2 lectures.
Wow. Can you hear me?
How about now? All right. We are all together. I'm going to go then to an assessment of something that we'll call terra incognita, the small intestinal microbiota, and its role in the development of an enteropathy called environmental enteric dysfunction, and how that relates to undernutrition in children and their mothers. And then I'll end by a plea for the establishment of human microbial observatory programs. And I'll describe what I mean by that. So buckle up and we'll proceed. So I had mentioned to you yesterday that childhood undernutrition is a devastating, pressing global health challenge, the leading cause of death in children under 5, not due to food insecurity alone, lots of factors operating within and between generations. Undernutrition is an intergenerational global health challenge, with mothers who are undernourished having an increased risk of giving rise to children who have undernutrition. Current therapies have reduced mortality, but they haven't ameliorated the long-term sequelae of undernutrition, which include stunting, dysfunction at the level of the immune system, impaired neurodevelopment, and that the definition of undernutrition is based on anthropometry, typically, which is a coarse description of the biological state or states of children with undernutrition. And I quickly reviewed for you some of the jargon associated with anthropometry. There's a median value that is the reference value. It comes from a WHO multi-country cohort study of healthy children. And stunting is defined by the degree of deviation from this median value. So an length for age Z-score, if it's -2 to -3, you have moderate stunting. More than -3, more than 3 standard deviations, severe stunting. Wasting, an impairment in ponderal growth, defined by a WLZ score, a weight for length Z-score. MAM being the term used to describe a moderate degree of wasting, moderate acute malnutrition, a WLZ -2 to -3, and then SAM, the WLZ score worse than -3. But we really don't have a good idea of a way of comprehensively defining the biological state or states of these children. And if we're going to connect the microbiome to the various perturbations and host systems physiology, we really do need to have a very comprehensive view of the biological state of these children. The central hypothesis I described was that there was a definable normal program of assembly of the gut microbiota, and that we had hypothesized that this assembly process, this microbial succession, was perturbed in undernutrition, and such perturbations were contributing causes to undernutrition, and that there was a necessity for proper co-development of the microbiome and host organ systems and subsystems in order to have healthy growth. I described a way of defining normal. We did a birth cohort study in an urban slum in Bangladesh, the capital of Bangladesh, Dhaka. The slum is called Mirpur. We took those children who had monthly anthropometry and monthly fecal collections beginning at 1 month, extending through 16 months of age. Did culture independent analysis, initially 16 sRNA amplicon sequencing, identified which bacterial taxa were present in those children who had consistently healthy growth as defined by anthropometry, used machine learning algorithms to identify age discriminatory bacterial taxa. This is a feature reduction exercise. And based on the proportional representation of these age discriminatory strains at different stages of postnatal life, we were able to define what should be the normal configuration of a microbiota. Many of these taxa are represented in children who are deemed healthy based on anthropometry living in other low and middle income countries. We further did feature selection by looking at covariing taxa and had a small group of organisms whose covariation changed consistently over the first several years of postnatal life. Normal community development is largely executed in the first 2 years of postnatal life, and that children with undernutrition have microbial communities based on the representation of these age discriminatory taxa that looks different, more immature than their healthy counterparts. And this immaturity is not, or this perturbation is not repaired by current therapy because none of these current nutritional interventions are based on knowledge or consideration of the developmental biology of the gut microbiota or microbiome. I use the term microbiome, by the way, to refer to the collection of genes in the genomes of organisms that comprise the microbial community. And I use the term microbiota to refer to the organisms themselves. And I know that's somewhat controversial, but it helps a little bit in the way we think about our approaches and what we're actually querying. Anyway, so these children have a persistent abnormality in one of their organs that happens to be a microbial organ. The question is whether that's a cause or an effect. We do a test of causality using notobotic animals, introducing well-preserved fecal samples collected from these children living in Mirpur, Bangladesh, in the Mirpur slums of Dhaka, Bangladesh. The key thing was we have protocols where we immerse the fecal samples at cryogenic temperatures in these charged shippers, that's minus 140 degrees, within 10 to 15 minutes after they're produced by the children, so that we can preserve not only DNA and RNA, but also metabolites. And these well-preserved samples, which are maintained subsequently at minus 80 degrees, and the informed consent is very important here. That's a whole issue that I'd love to be able to discuss later on with you. The ethical conduct of these types of experiments, ownerships of organisms, of microbial samples, the idea of trying to develop a narrative because of trusted relationships between the healthcare providers and the population that deals with the issue of future use of these samples. That's all inherent in the design and execution of these studies and the ethical conduct of these longitudinal studies. Be that as may, these well-preserved fecal samples from donors that are deemed healthy and donors that have undernutrition, chronologically age-matched donors, are each put into the intestines of germ-free animals. All the animals are fed a diet representative of the population that we're studying. The ingredients in the diet are cooked according to the cultural traditions of that population, irradiated and then fed to the mice. And we ask then, how much of the biological properties of the undernourished child can be transmitted via their microbiota to the recipient animals? And a variety of different aspects of host physiology are queried in these types of experiments. And it turns out that many of the features, including reduced lean body mass gain, alterations in bone biology, alterations in metabolism, effects on immune function, are evident in the mice that receive the undernourished donors' microbiota compared to the healthy controls. Machine learning algorithms are then used to query all the transplanted communities from all the donors in these animals to determine whether there are identifiable groups of taxa whose representation are correlated with different aspects of growth. These are deemed growth discriminatory organisms. They are then examined in the context of the microbial communities of the populations of children who are healthy or undernourished. And we see these growth discriminatory organisms are not represented in an age-appropriate way in the communities of children who have growth faltering. They become our therapeutic targets. The therapeutic approach that we use is one that has to be culturally acceptable, affordable, and scalable. And therefore, it wasn't a second generation or new generation probiotic consortium of these growth discriminatory strains, which are anaerobe, difficult to formulate, cold chain, not realistic to disseminate in a scalable fashion. The regulatory pathway for approval, complex. So we turn to the foods that are consumed by these children, understanding that every single family in this world has to make a decision about what to feed their children between the interval between cessation of exclusive milk feeding and a fully weaned state, this period of complementary feeding. And the guidelines or policies related to complementary feeding right now are not based on knowledge or consideration of the developmental biology of the gut microbiota. So we mine these complementary foods that are consumed by this population in experiments that involve the representative animal models that incorporated the microbes of the very population we eventually want to treat. And these microbes were initially represented as defined consortia of age and growth discriminatory strains that we had cultured, whose genomes we had sequenced. And the animals are then given a base diet, representative of the diet consumed, plus one of these complementary foods. And these complementary foods singly were examined for their capacity to initially increase the or effective fitness of targeted organisms. If we got hits, we would ask whether they had organoleptic properties that would be satisfactory, because we wanted to make therapeutic food formulations that would consist of combinations of these complementary foods that would hit our therapeutic targets, but also would be acceptable to the children who consumed or were given these therapeutic foods. In any event, we did this screen initially with the defined community, as I said, looking for hits, single ingredients, then multiple ingredients, and then took prototypes of these microbiota-directed complementary foods and tested for their ability to repair the microbial communities, the intact, uncultured microbial communities of children with undernutrition who have presented and been given standard therapy, but were left with persistent abnormalities in their microbiome configuration, and see whether those MDCS, microbiota-directed complementary foods, could repair these post-treatment microbial communities. In fact, among the MDCFs that we tested, several did efficiently. They affected different aspects of host physiology in ways that we deemed to be beneficial, but we were hesitant to go forward and directly test those lead MDCFs in children because we were trying to alter the developmental biology of humans. We didn't know about the safety because mice don't look like humans in a number of ways, physiologically and metabolically. So we took a deep breath, which had supplemental oxygen in that breath, and spent 2 years developing protocols for rederiving by cesarean section piglets as germ-free. The transfer from the sow to a notobiotic isolator done in a way where there was no environmental microbial contamination. And again, the introduction of the microbial populations from the very individuals who we eventually want to treat into these notobiotic piglets, and the testing of lead MDCFs coming from our mouse models. And voila, at the end of the day, we were able to get lead MDCS. We tested them initially in a pre-POC study, children who were 12 to 18 months of age who had MAM living in Mirpur, looking at the effects on microbiota configuration using those eco-group taxa as a way of defining the configuration, and the plasma proteome as a way of looking at the health status in a more comprehensive way of these individuals. Initially, 1,305 proteins representing biomarkers and mediators of different aspects of physiology. Took a lead MDCF from that, which actually was the highest performing MDCF in our preclinical model, and tested that in a more powered trial of children who presented with MAM, moderate acute malnutrition, who were 12 to 18 months of age, lived in Mirpur, that urban slum in Dhaka, and for 3 months, gave them twice daily either MDCF2 or a ready-to-use supplemental food, which is more or less the standard of care. MDCF had 15% lower caloric density than did RUSF. MDCF had these microbiota-directed complementary foods. In any event, the study design was serial collection of fecal samples, serial collection of plasma, measurement of 5,000 plasma proteins at time 0, time 1 month, and a time end of treatment phase, and a 2-year follow-up to determine what the long-term effects of treatment might be. And during the 2-year period of follow-up, every 6 months, a fecal collection, anthropometry, and a plasma collection. Primary endpoints for this study initially was going to be WLZ changes. Secondary outcome measures would be microbiome repair and changes in the plasma proteome. So MDCF2 had an effect on WLZ, the measurement of pondral growth, that was superior to that of RUSF, even though its caloric density was lower. We were able to correlate all children in both arms of the study, the effect of treatment on levels of these plasma proteins. The first thing we did with the plasma proteins was to bin them into those that were significantly correlated with WLZ. So all children in both arms, each one of the 5,000 different proteins, levels before, at the end of the 3-month period of treatment, how did that correlate with WLZ? Did we find proteins that were positively correlated? Yes, 70 of them. Many of them involved in musculoskeletal development, a number of them involved with CNS development. Those that were negatively correlated were involved mainly in immunoinflammatory responses. MDCF2 produces a significantly greater increase in those proteins in the plasma that were positively correlated with ponderal growth and a significant decrease, greater decrease in those proteins that were negatively correlated. So that's where we were. And I mentioned to you briefly that we've tried to look at structure-activity relationships in MDCF. We did mass spec of the ingredients in MDCF2, the ingredients in RUSF, found polysaccharides that were present in significantly greater levels in MDCF2, both the ingredients and the final formulation. We used those structures and then assembled MAGs, metagenome assembled genomes, from shotgun sequencing of serially collected fecal samples from all children in both arms, did the assemblies for each child, and dereplicated that. 1,000 MAGs of high quality were identified, looked at the encoded metabolic pathways, those MAGs, found that the positively correlated WLZ MAGs were enriched for pathways related to carbohydrate metabolism, looked at the expression of these pathways involved in carbohydrate metabolism, and found that there was a correlation between WLZ response and expression of these genes and carbohydrate utilization pathways, and that there was also a correlation between WLZ responses and levels of the microbial products of metabolism of polysaccharides present in MDCFs. To make a long story short, a lot of these transcriptional responses involved in polysaccharide metabolism, particularly the polysaccharides that were present in greater levels in MDCF2 compared to RUSF, came from two strains of Prevotella capri, and they were derived from polysaccharide utilization loci that contain genes encoding carbohydrate active enzymes that had predicted or known specificities for these particular polysaccharides in MDCF2. That's the way we got to the point of saying these are the bioactive components we think are really driving the ponderal growth response in MDCF2, and these are the responsive organisms. So I left off with this plea to ourselves and to you, which to ask the question, how well does this approach generalize to other populations? Populations that have different ages than the ones we tested. We tested 12 to 18 months, but remember the central hypothesis, the co-development of the microbiome and host systems and subsystems was important to healthy growth. Would it be as effective, with it being the MDCF2, if it were administered to a 6-month-old compared to a 12 or 18-month or 24-month-old child? Equally important, how about the timing of initial disruption of microbial community assembly, the duration of disruption, the severity of disruption of assembly? How malleable would the system be if you present to a healthcare provider with undernutrition and disturbed microbiome? Would disruption at 6 months make the system more or less plastic than systems that were disrupted, or microbiomes that were disrupted at 12 or 18 months? And again, the severity of the disruption. So we've gone ahead and done a couple of additional trials, one reported recently in Science Translational Medicine. The design of the study was identical to that applied to children with primary MAM. By primary MAM, I mean children who present with moderate acute malnutrition. There's no antecedent history. In particular, there's no antecedent history of SAM, severe acute malnutrition. And by the way, I whispered to you that the World Health Organization has no policies, formal policies about treatment for MAM, in part because current therapies don't have an effect on the long-term sequelae of MAM. And although there is a protocol for treating SAM with ready-to-use therapeutic foods, typically those children are treated with these ready-to-use therapeutic foods, which are not designed with knowledge of or consideration of the developmental biology of the microbiome until they reach a state of MAM. So they have post-SAM MAM. But again, they have the long-term sequelae. So we took children living in Mirpur, that urban slum, as well as in a rural area of Bangladesh, Kurigram, who had post-SAM MAM. And they were treated initially with standard therapy for SAM. They were left with MAM. At the time they achieved a state of MAM, they were given either MDCF2 or RUSF for 3 months. The same serial sampling of the microbiome, the same serial anthropometry, the same serial blood draws, time 0, 1 month, and 3 months. And again, we're followed for 2 years. I also emphasized to you yesterday, that the WHO and all healthcare organizations have had no policies described for, and really no effective treatments for stunting. Okay, that's really been very recalcitrant. This particular study yielded a result that was very similar to what we got with children with primary MAM. The ponderal growth response was significantly greater with MDCF2 compared to RUSF in these children with post-SAM MAM, even though MDCF2 had lower caloric density. It's not just calories, something much more profound going on. The 2-year follow-up, just as it occurred in the case of primary MAM, at the end of the 3-month intervention, there were no statistically significant differences in linear growth responses. Those became evident during the follow-up period of time. And again, significant improvement with MDCF2 compared to RUSF in linear growth. And just as we saw with the primary MAM study, the most significantly associated MAG with WLZ were P. copri, belonging to P. clade A, that had a unique collection of loci involved in the degradation of polysaccharides represented in MDCF2. And also there was another organism that was significantly positively correlated with WLZ in this particular study, and also in the primary MAM study, Geminger formicillus. And this is a diagram developed by Steve Hartman, who is a graduate student in the lab, where you look at the representation of metabolic pathways in these 2 P. copri that were WLZ associated. There are other Prevotella that are present in the microbial communities of these children, both in the primary MAM study and the post-SAM MAM study, that were not associated with WLZ, and also lacked the full ensemble of loci that are involved in the degradation of polysaccharides that we think are the bioactive component. You can also tell from this wheel diagram the representation of pathways for amino acid biosynthesis. Note that those MAGs that are positively correlated with WLZ, the P. copri MAGs belonging to clade A, Geminger formicillus, have all the pathways for amino acid biosynthesis represented. In any event, the long and short is that we can correlate the responses of these children to the representation of MAGs with particular genomic features related to polysaccharide utilization, and MAGs that are not associated with ponderal growth responses that lack these particular features. And that, I think, is very helpful for us in terms of prediction of which children might be responsive and not, and also dealing with structure-activity relationships, both at the level of the therapeutic lead, MDCF2, and also structure-activity relationships in the microbiome, and the importance of considering strain-level variation and understanding the response. Now, imagine if you're doing somologic-based analysis of 5,000 plasma proteins in all of these studies. We can identify, as I told you before, using an approach of correlating levels of plasma proteins, the change in levels of plasma protein as a function of treatment with LAZ or WLZ responses. So we identify a whole series of proteins that are LAZ associated. And we do so in the case of LAZ-associated proteins in both studies. And now we start assembling a series of biomarkers of treatment responses and biomarkers that could be used to stratify population. As in the case of the primary MAM study, there's a significant effect on linear growth that's not evident in the first 3 months of the study, but is evident in the period of time that elapses afterwards. The importance, again, of being able to do longitudinal follow-up and the commitment of the healthcare providers and the participants and the participants' family to these types of studies to improve the health and well-being of the population. So this is just a quick overview of some of these LAZ-associated proteins that were identified in these studies, and their linkages to musculoskeletal development, energy metabolism, CNS development. What's very profound is the fact it's well known that there's a relationship between stunting and CNS development. Impaired linear growth is associated with impaired central nervous system development and cognitive development. So what are the effectors of these processes and how they might be shared? And you can see from this particular slide that these LAZ-associated proteins that are involved in musculoskeletal development include those that are participatory in bone mineralization, extracellular matrix formation and maintenance, the remodeling of the matrix. In the case of the CNS, there are proteins that are associated, positively associated with LAZ, that are involved in neuronal development and axon guidance, in neurotrophic signaling, in synaptic transmission and plasticity. Again, these are proteins that we can detect in the plasma. There's a whole other interesting thought about how these biomarkers that are parked normally in the CNS become detectable in the plasma. But I think somologic technology is quite robust, and these are an invaluable set of biomarkers and mediators that we are applying not only to our studies of humans, but bringing back to our mouse models. Remember, I told you last time that we can take the pretreatment samples from the participants in these studies and introduce them into our notobotic animal models, reenact the trial. And typically, these mouse models contain defined consortia of age and growth discriminatory strains. We can manipulate the representation of those strains by including them or not including them, and really trying to understand which strains are associated with what type of responses that are seen in the human population. Connecting the dots in this virtuous cycle of doing a clinical trial based on preclinical models that are representative of the population, taking an investment in mechanistic analyses of the participants in the clinical trials, and then going back into a preclinical model and further exploring those sorts of responses, but at a level that is not possible in human population, to better understand what kind of biomarkers we should use to assess treatment efficacy, whether we can do adaptive clinical trials in the future, and more importantly, whether we can stratify populations based on these biomarkers, so we understand better how to treat, how long to treat, what doses to give, et cetera. So, the other thing I wanted to mention about tests of generalizability before I go into terra incognita, small intestinal microbiota, is this Nutrimam study that's being supported by the Gates Foundation and the World Health Organization to test the effects of MDCF in 6 to 24-month-old children with MAM living in Bangladesh, India, Mali, Pakistan, and Tanzania. Basically, it's a bake-off between RUSF and MDCF and locally available foods, a 3-month intervention, and then a 3-month follow-up, not 12-month, unfortunately, or 24-month, just to determine whether the intervention has similar efficacy in 6-month-old children with MAM, or 12-month-old or 24-month-old children with MAM living in a given locale and in different locales. In each of those locales, we've had to do a birth cohort study to identify what is normal. That is to say, you do a birth cohort study, you do serial anthropometry, at least over the course of the first 24 months of life, take those children and have consistently normal anthropometry, and then do culture-independent analysis to identify the representation of different taxa at different stages of development. Use machine learning to identify age discriminatory strain. And that's the reference by which you judge the efficacy of microbiome repair. We want to make sure that we take into account some geographic differences in the features of postnatal development of the microbiome. Everybody follow me at this point in time. Deep breath. Pause. You can all stand up, stretch, etc., whatever you wish. And I want to talk about the small intestinal microbiota and the work of Kaylee Bruce, Clara Cow. Kaylee is a postdoc in the lab who did her PhD with Justin Sonnenberg. Clara is an MD PhD student in the lab who just graduated. Zinan Chang is in our post doctoral medical scientist training program. He did his fellowship, clinical fellowship in gastroenterology, and Rand Koskin, who is a very talented MD PhD student in our immunology program. So this project is focused on the mothers. Remember I said there's maternal transfer of microbial community to the offspring, and we're very interested in the role of the maternal microbiome in the pathogenesis of undernutrition, not only in women, but in their offspring, the intergenerational perpetuation of risk for undernutrition. Stunting affects about 150 million children under the age of 5. It's a chronic manifestation of undernutrition. Remember I said there are links between stunting and impaired neurodevelopment. There are metabolic and immune dysfunctions also associated with it. You see the distribution of stunting around the world, color-coded in this fashion. I wish we had more time. We could talk about this at greater length. But there is an entity that many people were interested in for a long period of time called EED, environmental enteric dysfunction, first described in Peace Corps workers who came to the United States after being in various low and middle income settings, particularly settings where there was high levels of fecal oral contamination. They came with histories of weight loss, diarrhea, and malabsorption. When they returned to the United States, they were endoscoped. And what was found was a disruption of epithelial barrier function and loss of the absorptive surface of the small intestine manifested by collapse of villi. Fewer villi, not as high. This villus atrophy, loss of absorptive surface area, disruption of the barrier with associated immunoinflammatory responses in the lamina propria, had an etiology that was unclear. Typically, there was no concerted effort to treat in a single way. There was no protocol. And these Peace Corps workers would typically, over time, recover their absorptive function, their histopathology would resolve. Was this abnormality something that was present in undernourished children? Was this abnormality present in mothers who were undernourished? There were no validated biomarkers for EED, particularly in children, because from an ethical standpoint, it was not possible to do endoscopy on healthy children living in a particular area. And the risks of doing endoscopy in a resource-poor environment in children who are undernourished was considerable. You had to have a rationale for doing that. So the Gates Foundation said, enough is enough. We don't have gold standards for diagnosis. Let's do endoscopy, much like was done with the Peace Corps workers in children who failed to respond to a standard intervention for undernutrition. This particular case, a study was done with our colleagues at the International Center for Diarrheal Disease Research in Bangladesh, involving children who presented with stunting and who were 12 to 18 months old. There were 525 children. They either were frankly stunted, that is to say their LAZ was less than 2, or they were deemed at risk for stunting because their LAZ was less than -1. There was a 3-month intervention, an egg and milk 6 days a week with micronutrient sprinkles. And among those children, some failed to respond. Remember, I said there has not been much success in surmounting the problem of stunting. And there was a superb group of endoscopists, gastroenterologists in Dhaka associated with ICDDRB, and the Gates Foundation felt that the safety of endoscopy and the ethical justification for endoscopy were in place so that a study could be done to determine whether these children who failed to respond had EED, environmental enteric dysfunction. And we would determine that by doing a mucosal biopsy from the duodenum, obtain the upper endoscopy. Duodenal aspirates were obtained so we could culture the organisms present in the proximal portion of the small intestine. Fecal samples would be obtained, as well as plasma samples, so we could apply the somologic method of aptamer-based proteomics. So the net result was that matched plasma and duodenal biopsies were obtained from 84 of these children. 80 of the 84 had biopsy-confirmed EED. So these children that failed their nutritional intervention, the vast majority had EED. And we got duodenal aspirates for culture from 38 of these, and culture collections were generated. And this was done by Robert Chung and a very talented postdoc working with him. And 2 culture collections were generated, a complete community, that is to say, all the cultured organisms that we could get a hold of that have been clonally arrayed, and then a subsetted version where we took one strain that represented the species that are represented in the culture collection, so a reduced or subsetted community. And those defined consortia were introduced into female mice who were fed a diet representative of those diets consumed by adult women living in Mirpur, where the study was done. Again, food, the ingredients cooked according to the cultural traditions of the area. The food formulation was irradiated, pelleted, and given to the female mice. Then these mice were mated, and their offspring were weaned onto a diet representative of that consumed by 18-month-old children living in Mirpur. The question was, did the complete community, did the subsetted community produce pathology in the small intestine that resembled the pathology that was observed in the children who had EED? And the control was to conventionalize, take an intact microbiota from the cecum of conventionally raised animals and introduce into the mothers prior to mating. Well, amazingly, the complete culture collection produced systemic inflammation as defined by levels of lipocalin in these mice. I'm going to go through this inflammation beginning at the level of the small intestine. So within the small intestine, in both the duodenum, the ileum, and the colon, levels of lipocalin-2 or LCN2 were significantly higher in mice with the complete community compared to the subset community, and also compared to the conventionalized animals. Levels in the gut of CD3 positive cells, Th17 positive cells, neutrophils were higher in the animals that have been colonized, the offspring of the dams that have been colonized with the complete community compared to the subset community. And there was abnormalities in cryptbillus morphology with billus blunting. Serum levels of lipocalin also were significantly higher in the offspring that had received the complete community from their mother. There is evidence of systemic inflammation as judged by increases in levels of splenic neutrophils. There was increased levels of neutrophils in the meninges. And we decided to name the different consortia based on this observation of local and systemic inflammation. In the case of the complete community, it would be called CSII, or child small intestinal inflammation inducing bacterial consortia, and CSIN, the subsetted community, child small intestinal non-inflammatory consortia. So what does it look like in the gut? So single nucleus RNA-seq was performed by Zenon, and then pseudobulk analysis performed to look for differentially expressed genes. And I don't have enough time to go through this in detail, but I'll give you a high-level overview. Expression of genes was significantly higher in enterocytes that are associated with barrier function, components of tight junctions, marked alterations in energy and lipid metabolism that was evident in recipients of the CSI and the consortium compared to the complete consortium. And then in the case of offspring of dams receiving the complete community, the inflammation-induced community, leukocyte genes involved in leukocyte migration, phagocytosis, efferocytosis, which is an interesting process involving clearance of dead cells and debris, all increased. If we take a look at signaling between cell populations, we do niche net analysis of our single nucleus RNA-seq data set. Niche net allows you to designate defined populations of cells, cell clusters, as sender populations, and other cell clusters as a receiver population. And you basically look for ligand receptor interactions that are well known. So we can use these types of analyses to find that there's increased signaling from Paneth goblet cells to stem cells in animals that receive the CSI, the inflammation-inducing consortium. And these pathways are pathways involved in Wnt, BMP, and Notch signaling. These are the three major pathways that mediate cellular proliferation during normal development, during injury responses, and during responses to inflammation. This abnormality in host biology in response to the CSII consortium extends to the brain, and there are interesting abnormalities that we can detect, Cayley detected using single nucleus RNA-seq. And that involves genes involved that participate in angiogenesis, genes that are involved in myelination. The inhibitors of myelination in the brain are increased in CSII mice. There are pro-inflammatory cytokines that are affected. Their increased expression is evident in these animals that receive from their dams the complete community, IL-15 in microglia, IL-18 in astrocytes. There are genes that regulate growth and synapse formation, things like neuroregulin that are diminished in CSII. And then Ellie Byrne, who is a graduate student lab who just finished up, adopted for notobiotic isolators the capacity to look at behavior of pups and their dams in the first 2 weeks of postnatal life. Doing these sorts of studies in notobiotic isolators is cumbersome. These behaviors included reflex development, but also the vocalizations that occur between the pups and their mothers. That's measured by ultrasonic vocalization recordings. So this is done in a multi-generational model where you have lines of mice that have these consortia, two different consortia. So those animals that have received, the pups that have received the inflammation-inducing CSII consortium have impaired reflex development and increased ultrasonic vocalization, a response that would be indicative of stress. So behavior is also affected. Now, which microbes might be responsible in this consortium for the immunoinflammatory response? One way of doing this is to do co-housing experiments. So you have mice that either receive the CSIN, the non-inflammatory, or CSII, inflammatory consortia. Control animals are just colonized and not co-housed. And then the experimental groups are co-housed, either animals that first received the CSIN and then are co-housed with the CSII, or animals that first receive the CSII and then are co-housed with the CSIN. And the answer to that sort of analysis of whether there are organisms in the CSII consortium that could invade and produce inflammation in CSIN, the answer was yes. In fact, that does occur. You can see initially that based on serum lipocalin 2 levels in the upper right-hand corner. If you take the non-co-housed control, CSIN, or the non-co-housed control, CSII, depicted as blue and red, you can see a difference in the levels. But take a look at the co-housed animals. If they start out as CSIN and were co-housed with CSII, the levels of lipocalin 2 in serum increase to those documented in non-co-housed CSII, and the same thing that would happen with CSII that were then co-housed with CSIN. So this idea of there are elements of the CSII consortium that could affect the properties of the CSIN, were also manifest when we took a look at levels of CD4 positive cells in the small intestine, neutrophils in the small intestine, neutrophils in the spleen, and neutrophils in the meninges. So then the fun begins. Did a MAG-based analysis, even those who find collections of microbes. And this is a complicated heat map. If you take a look at the bottom, it deconvolutes all of this data, so you don't have to get indigestion looking at this thing. Basically, what we have are it's a heat map of pairwise comparisons of the abundance of different bacterial strains, the differences between non-co-housed control, which is on the left, that left block, or co-housed groups compared to CSIN, that's in the middle. So all co-housed versus CSIN non-co-housed, or to CSII colonized animals, all co-housed on the right compared to the control non-co-housed CSII. And these different cells in this matrix are subsetted to those bacteria that are significantly different in abundance. And the significant difference in abundance is indicated by the star in at least one comparison. At the end of the day, after looking over this data, you come to the conclusion that there are 3 bacterial taxa that are elevated in co-housed animals compared to animals that have the non-inflammatory CSIN. And those organisms include Campylobacter concisus, which is a member of the oral microbiota, well known as a member of the oral microbiota, and also several strains of Actinomyces, Donoliticus, and one other strain. Those are the red arrows. So what do you do with this information? You do add one back experiments. So remember, this is a culture collection. This is the power of having a clonally arrayed culture collection from the get-go. So you colonize mice with the CSIN non-inflammatory consortium and add to these animals after a period of initial colonization, either the complete consortium of 2 Actinomyces in the C. concisus, or just the C. concisus. You can do the same thing with the individual Actinomyces as well. And the net result of these add-on experiments, and you could look at the middle panel where the levels in the colon of LCN, lipocalin-2 are measured, is that when you add to the CSIN containing mice, all 3 organisms, there's an increase in lipocalin levels. This increase is also seen with concisus alone. So this gives us an idea that C. concisus is an actor that's producing mayhem, that's producing an immunoinflammatory response, and it is illustrating a concept that's emerging with this pathology, that there may be a decompartmentalization of the microbiota, and organisms that live normally in one portion of the alimentary tract are getting established in an area where they shouldn't be and causing pathology. And if you want to go further, and I don't have time because we're almost up, and Dr. Ray has a taser, which he promises to use on me if I become too verbose. This just shows an analysis, a bulk RNA-seq analysis that reveals that there's increased expression of immune response, inflammation signaling, and cell death pathways in the distal gut of animals gavaged with concisus. This is an add-one-in experiment where the initial colonization is with the CSI and non-inflammatory consortium. So we get to an idea of what pathways are modulated. So have an intergenerational model, characterize the effects of these 2 consortia, culture from duodenal aspirates obtained from stunted children with EED who failed a standard nutritional intervention. We see that there's a consortia. The small intestinal microbiota of these children contains organisms that can induce systemic inflammation. The inflammation extends beyond the walls of the gut to affect things like meninges. There are defects in gut barrier function that we can identify through a variety of means, including RNA-seq and more directed measurements. And also there's an effect on brain development that we see. The co-housing gives us an idea of which organisms are maybe mediators. The interesting thing at the end of the day with these experiments was that Rhiannon looked at intrauterine development. Now, you know that IUGR, intrauterine growth restriction, is a huge global health problem. And we think about postnatal development being impaired. How about intrauterine development? So if you take a look at the fetal weights at E11.5 is when placentation just is completed. In a later stage of pregnancy, you see the fetuses have lower weights if the dams are colonized with the CSI-I consortium. So there's an effect of the small intestinal microbiota on intrauterine growth. And how might that be coming about? So back to the drawing boards, and there are women, adult women in the Bangladeshi EED study who came in with wasting as judged by low BMI. So there are groups of women, reproductive age, 18 to 45, living in Mirpur, who were defined as undernourished, as with a BMI less than 18.5, and healthy with a BMI greater than that value. Those that were undernourished were subjected to an intervention similar to that applied to children. The non-responders were given an option of endoscopy to determine whether they had EED. But in this particular case, we had healthy women who had been referred to the GI clinic for functional dyspepsia, who didn't have evidence of celiac disease. And we, unlike with the children, could obtain mucosal biopsies, duodenal aspirates, fecal samples, and plasma samples for healthy controls. So this was key. We wanted to determine whether EED was associated with undernutrition in adult women living in this area, because we were wondering whether there might be transmission of a EED-inducing consortium from women to their offspring. The non-responders in the group of women, there were 38 that were eligible for EGD. That's esophagogastroduodenoscopy, the aspirate and the biopsy. We confirmed EED in the vast majority of these women, and then went ahead and did an intergenerational transmission experiment of culture collections that were generated from women who were undernourished and culture collections that were generated from the duodenal aspirates of women that were deemed healthy. And the design was very similar to the one I talked to you about. Female mice, germ-free, put on an adult Mirpur diet, were colonized with one or the other consortia. They transmitted the consortia to their pups. The pups were studied initially at different stages of intrauterine development, but also at different stages postnatally. What happened in the dams with the adult small intestinal low BMI consortia, ASIL, my God, the jargon's getting ridiculous, had compromised barrier function, and that could be measured by serum levels of phytidextran, increased systemic inflammation. We did single nucleus, Xenon did single nucleus RNA-seq and bulk RNA-seq. There are all sorts of perturbations that were identified. There were discordant phenotypes in the offspring of dams colonized with the consortium. We're now looking at different stages of postnatal life, and you can see that there are lower weights that emerged in offspring of dams that had the consortium from the low BMI or undernourished women compared to the healthy women. There are increased levels of serum inflammatory markers, in this case, LCN2 and matrix metalloproteinase 8. And there's also an effect on bone biology with diminished trabecular mineral bone density in those pups that had inherited the consortia from the dams that originated from undernourished females. We then turn, we being Rhiannon, to the mouse placenta, wondering what the effects might be on placental and fetal development. Just a quick review. The maternal component of the placenta is a decidua. It's a modified endometrium. There are two zones that are the fetal component. The labyrinth zone separates maternal blood from fetal blood cells. It's composed of centrophoblasts and cytotrophoblasts. There's a junctional zone, glycogen-rich cells, trophoblastic giant cells that maintain the maternal-fetal interface. There's a particular resident natural killer cell population in the deciduized endometrial component of the placenta. And these are very important. The uterine NK cells perform critical roles in spiral artery remodeling during healthy placental development. In order to get a good vascular system installed in the placenta, you need these NK cells to function properly. So what was seen by Rhiannon and her indicted co-conspirators is that placental weights were significantly lower early in pregnancy in mice colonized with the low BMI donor microbial consortium. And these reductions in fetal weight were, these placental weight effects were also accompanied by alterations in fetal weight. And to make a long story short, single nucleus RNA-seq of differentially expressed genes in these uterine NK cells that was performed at E11.5, which is the end of the period of placentation, indicated significant alterations in NK cell function. That includes effects on expression of genes that are involved in infiltration of these cells, their cytotoxic granule and interferon gamma production, their activation, and evidence of an exhaustion signature. So there's clearly a connection between the consortium that's introduced into the small intestine and these aspects of placental development. You can do co-housing experiments now of the type that I described now before, where you take mice that are initially colonized with the consortium from the healthy donors, the consortium from the low BMI donors, and after a period of time, and this is done prior to mating. So these are the dams that are being colonized with one or the other, or are cow-housed after initial colonization. What's the effect on placental and fetal development? It turns out that there are organisms in the healthy consortium that when introduced into the consortium from the low BMI donors can impact effects on fetal development, reverse the impairment in fetal development. So undernourished Bangladeshi women of reproductive age have evidence of EED and systemic inflammation. Mice colonized with the consortium collected from healthy women, and compared to mice who have consortium from undernourished women, have alterations in placental and fetal weight. The reduction in placental and fetal weights observed in mice that have received the dams that have received the low BMI consortium. The NK cell dysfunction appears to be at least linked to this perturbation and vascularization. And we're using notobotic mouse models to define which consortia have infiltrated the low BMI collection from the healthy consortia as next generation therapeutic targets. Who's willing to spend 5 more minutes, and I'll talk briefly about microbial observatory programs. But it is quite remarkable, we think, that the small intestinal microbiota of women who are undernourished could impact intrauterine, placental, and fetal development, and how when transmitted to offspring, they also affect postnatal development. And the idea that there may be a target that could be modifiable that would affect growth prenatally and postnatally. So that's very attractive. And the types of approaches to identify therapeutic leads are those that I described here, with co-housing, add one in type of experiments, and then therapeutic lead development. You have the targets, then the leads in these representative preclinical models, and then the application of leads to the human population whose microbes are used to develop the preclinical model. So last point. I think it's very important to behold where we're at right now in life on planet Earth, and to realize that there is a century of profound change that is occurring and will continue to occur, both in terms of our environment because of climate change, our population of humans living in different areas of the world with geopolitical disruptions, increased gradients in wealth, changes in the epidemiology of different diseases. So it could be not only timely, but essential to develop human microbial observatories at sites in selected low and middle income countries where long-term sampling of microbial ecology could be conducted. But to do that in a way that's thoughtful, in a way that is ethical, and a way that is more economical, is not to do all by all, but to think deeply about what sites you would want to choose and what are the parameters for selection of such sites and what are the architectural features of such observatory programs. So I just want to throw some ideas out at you. Site selection. Perhaps the key thing would be that the population exemplifies a global health challenge or challenges of interest, that the anthropologic features of the population are such that they represent a population at risk for a disease or disease states, and that these features are predicted to change over the next several decades. So too, sites where not only the epidemiology and anthropologic connections are expected to change, but the environments in which the population lives are expected to change, in part because of climate change, and how these changes would inform both the environmental and human habitats that would be sampled. It could be or should be sampled. Community engagement and trust. Absolutely essential that the level of community engagement and trust be great. The trust is between the healthcare workers and community leaders, and it's based on a sustained effort at communication. Without that trust, you're never going to be able to do longitudinal studies. And that includes birth cohort studies, as well as interventional studies where long-term follow-up is required. Deep, deep knowledge of how government regulatory policies will impact microbiome research, both now and likely in the future. And that has to do with policies related to sharing of biospecimens, policies related to approval of pre, pro, or symbiotics, combination of pre and probiotics, and also policies related to the distribution within and across national boundaries of microbiome-directed therapeutics, including foods. Another consideration, what are the best practice policies for obtaining ethical consent? That has to do with a deep consideration of policy related to ownership of microbes, NAGOTA protocols, as well as future use provisions of future use, not only for non-commercial purposes, but also for commercial purposes. And that also entails, under that umbrella, good policies, equitable policies, realistic policies for obtaining economic benefit for local and national industries. That's good stewardship of IP, and also communication between the scientists and the healthcare workers and governmental agencies, so everybody is educated about intent. Decision-making tree about which body habitats should be sampled at what ages, and how the sampling efforts relate to diseases of interest. So you need an infrastructure for preserving biospecimens. You collect the biospecimens, but how are you going to preserve and protect them in terms of the viability of the organisms themselves, but also the metabolic products, and how you're going to retrieve these specimens in a cost-effective way. And to do so, I think a careful alignment with the diseases of interest and how many partnerships you have or imagine you could obtain from both commercial and non-commercial sources. And everything is linked in part to a consideration of what kind of therapeutic development programs you might be able to initiate. Probably best to brand these observatories as regional centers of excellence for capacity building. So that site becomes a site where technology is built up and where knowledge and expertise is built up by training individuals to do these types of studies. And these studies can radiate their expertise to other sites within the country and even across national boundaries. Policies about data exchange between centers, and also the ability to harness these observatories as sites for doing clinical studies. So single center or single site studies, but also multi-site studies. And then finally, they should be branded as decadal programs because this change will occur rapidly, but over the course of decades, and you need sustained funding. And I think one way of obtaining such funding is linked to programs related to climate change and environmental sustainability. So thank you for your attention the last 2 days. That's our story. We're sticking to it and really appreciate all of your efforts to listen to the work of some very, very talented young people shown here.
Questions?
Thank you, Jeff, for your thorough presentation. I have two questions. One is the food composition of the MDCF2 was determined based on the fact that it was readily available and culturally acceptable in the context of Bangladesh. So I'm wondering whether it is applicable for the other parts of the world, because I'm thinking that the gut microbiome composition and function is supposed to be different for the other parts of the world in the children who suffer from malnutrition. This is number one question. Number two question is, I know that ED is causes the 40% of the malnutrition. So did you find any non-invasive biomarker that can distinguish between malnutrition with ED and malnutrition without ED? Because histologically, it is difficult to distinguish ED and malnutrition. So,
well, the first question about how generalizable will be the effects of our current MDCF formulations and other sites, that's what the purpose of the NutriMam study is. So that's ongoing. Equally important is how generalizable it will be across different ages at a given site and between sites. Each of the sites that I mentioned, there's a birth cohort study that's being performed or has been performed to define normal, and we'll just have to see. I also mentioned yesterday that the generation of shelf-stable therapeutic foods directed to microbiome, their distribution is a very large logistical problem. And ultimately, we would like to take knowledge of the bioactive components of microbiota-directed foods. In this case, I think a major component of the activity can be ascribed to polysaccharide, and to identify sources of those polysaccharides so we could obtain them at scale and develop a prebiotic cocktail that could be sprinkled onto food. A lot of the efforts in the lab is directed at that. That's question one. Question two is getting biomarkers from these types of studies that I just described that would allow us to identify individuals with EED, and specifically EED in forms that are associated with manifest undernutrition. So the approach that we're using now is to take our mucosal biopsies and do somamer-based or somologic aptamer-based proteomics to get a richer repertoire of proteins whose expression in the mucosa is correlated with the nature of the microbiota that we see is associated with pathology. So there are a specific set of organisms that we feel are associated with increased risk of pathology. How are these proteins correlated with that, and manifest pathology by histologic analysis, and also by classical measurements of nutritional status, including BMI. So that's just going to take some time to do, but that's what we're trying to do.
did you validate those biomarkers in the stool samples or plasma samples?
We're doing that now. That's an ongoing study. And you could appreciate the fact that it's not simple to do upper endoscopy. And I think buried in your question, but appropriately subsumed in the question, is are there other ways of sampling the small intestinal microbiota? And there are capsules that have been developed that can be swallowed for sampling. Those are still in the validation phase. I still think that it's going to be important to do endoscopy for a period of time to get an accurate view of the extent of histopathology. So there's lots of work to do, but I do hope that you'll take away from this the message that the small intestinal microbiota, a terra incognita, may play an important causal role in the pathogenesis of EED that contains therapeutic targets potentially that could be utilized to treat disease and ultimately present, and that the EED associated consortia not only has effects on postnatal development, but intrauterine development. And we've not published much of this yet. A lot of these studies are ongoing, but I thought I'd want to share that with you now, tell you what the vector of effort is. Ellen. That's not the only culprit. We think it was an exemplary case in the case of the child's duodenal culture collection.
Yeah, but I'm wondering if that is also true across different populations, and also is that would suppressing that bug be a good thing, like maybe with the bacteriophage strategy or something like that?
It's interesting that that's a member of the oral microbiota, and one wonders whether sampling the oral microbiota might be a good way of screening, going back to the question that was asked before, individuals who might have an associated enteropathy. We need to know more, as you imply very politely, about the representation of that organism in populations of individuals living in a given locale who have EED and across locales. And I don't know what the answer is going to be. Now, that was initially a study that started out in children. We've gone back to the mothers, and we'll probably spend more time looking at the organisms that ameliorate the pathology, including the intrauterine growth pathology, because we can consider the differences between healthy and undernourished duodenal communities, because we can do endoscopy ethically. And it would be nice to be able to find an organism whose representation and express function would treat and ultimately prevent disease. So that's probably the approach that, well, certainly the approach that we're going to devote our attention to now.
Any other questions? Great.
Shall we all rise for the benediction?
Thank you again, Jeff Gordon.